So in the space of immune therapy, I think it's very important to emphasize the excitement around adoptive cellular therapies, and in particular CAR T. And obviously CAR T therapy has proven curative in leukemia and lymphoma in selected patients. And in myeloma, whilst it remains to be approved from a regulatory standpoint, early results from a variety of trials are very promising. I think what's very important for patients to appreciate, though, is that CAR T therapy so far appears to be highly effective in highly selected patients. And the only challenge has really been that whilst the response rates are very high and the quality of response rates in those patients who can successfully undergo the procedure do seem to enjoy high rates of MRD negativity, as we call it. What's been somewhat challenging is that the duration of disease control has been relatively limited, about a year to a year and a half, to at best 2 years or so, depending on the quality of response. Now, I personally think that's very encouraging, because these are very sick patients who've had multiple prior lines of treatment and have become highly resistant, and most available therapies have failed them. So I think the signal in itself is very positive. I think exactly how this will then translate when it's used earlier is an area of very active study. But broadly speaking, CAR T therapy, I think, has really shown that it's got great potential. Our ability to then try and maintain it or maintain its benefit becomes a major next step. In this context, obviously, therefore, a very important piece has been to figure out how we can maintain the benefits that CAR T appears to engender and allow a greater and more prolonged period of disease control. Now, I think in doing that, we have to understand a lot about the biology of T-cell immunology in myeloma and how complicated that is. And also understand barriers to resistance, or rather barriers that are brought on by resistance, and what exactly are they. And I think one very interesting area has been this understanding that natural killer cells are very important. And so there's a whole new space of evolving adoptive cellular therapy using a natural killer cell platform as opposed to a T-cell platform. And so I think the important message for patients is that the field is really dynamic and moving, I think, in a number of very positive directions. But I think the most important thing, in my view, that needs to happen in the CAR T space is to get an element of scalability. Because right now, as I mentioned previously, the waiting lists for clinical trials are long. The ability to actually treat patients in a very timely fashion is always complex. So I think it's a very exciting area, very promising, but a lot to do. And that's why I think when we think of other BCMA targeting strategies, such as the GSK916 antibody drug conjugate, that's kind of remarkable, because that's an off-the-shelf antibody that you can simply just give. It's a once every 3-week infusion. There's some new schedules looking at once every couple of weeks to sort of reduce some of the side effects that are seen. And that's proving very, very interesting as well. So immune therapy, broadly speaking, very exciting. I think another area in immune therapy that's underappreciated is the role of actually the next generation immunomodulatory drugs. And by that I mean what we now call cell mods. They're called cell mods because they're really potent, but they're in the IMiD family. They target the cereblon ubiquitin ligase complex that controls the targeted degradation of Ikaros and Aiolos. And basically, this whole pathway has been much better understood, but it's also very complex too. But the really remarkable thing is that these next generation drugs, which build upon lenalidomide, build upon pomalidomide, are even more potent. And very, very critically, they work when both lenalidomide and pomalidomide have failed. The first one is iberdomide, IBA for short, and that has the number CC220. And there's a next generation one right behind it called CC480, and these two oral agents are really, I think, showing promise. The most mature data we have is on CC220, and they're about a third of patients in whom most other classes of drugs have failed them benefit from its use. And this is a simple pill taken daily, 3 weeks on and 1 week off. Very similar to pomalidomide, very similar to lenalidomide in that regard, but much, much more potent. So these are examples of a variety of immunotherapeutic strategies that we're exploring going forward. I think it would be very important, however, to say that beyond immunotherapy, there are a lot of other exciting things going on in myeloma, and I think there are a number of examples that are probably worth sharing. Beyond, obviously, the immune strategies lies the whole idea of other targeted approaches. And one of the most interesting and one of the most important in my view has been the emerging role of venetoclax, which targets the abnormality of translocation 11;14, and in particular mechanistically targets patients in whom their disease overexpresses BCL2. Now, what we've learned there is something of a cautionary tale, that if you don't have in your disease translocation 11;14 and or you're not overexpressing BCL2, venetoclax may not be helpful. In fact, it may be quite unhelpful. And that's been an important lesson. Having said that, if you do have 11;14 translocation in your disease, venetoclax can be remarkably helpful in combination with other drugs like, for example, bortezomib, and for that matter, carfilzomib. So I think that that's a very interesting example of a targeted approach going after a very specific cytogenetic abnormality. And I think in that same spirit, though, I think it's important to note that some of our better drugs in myeloma are very broad in what they do. And in that space, we've had a couple of really new and exciting developments. So in this particular area of targeted approaches that are non-immunotherapeutic in the classical sense, but exploit potentially novel mechanisms of action, I think there are a couple of very nice examples to share. One of the most recent FDA approvals has been for the nuclear export protein inhibitor, or so-called selective inhibitor of nuclear export protein, and that's selinexor. These are so-called SINEs. They target a particular pathway modulated by a very important exporter of nuclear material called XPO1. What these do is they export from the nucleus critical proteins that suppress the activity of specific oncogenes, their RNA products, and a variety of other effects. And this has been a remarkable breakthrough. It's a totally novel mechanism of action. And how selinexor appears to work, I suppose a good analogy to share with patients is this, that it's almost as if at home there's a rave going on and the parents have left. And what you do then to sort of shut this process down is you bring the parent back. So by inhibiting the export of the parent, as it were, keeping the parent in the house, the rave is converted to a sort of wine and cheese. And literally, in a very simplistic way, that kind of makes sense. Because what you do is you make the nucleus behave more appropriately for what's going on with its gene transcription products and so on. And as a result of that, actually, the myeloma dies. Because it essentially relies on these nuclear export protein mechanisms to survive, because obviously it's behaving aberrantly in a malignant fashion. So that's perhaps a rather simplistic explanation of what selinexor is, but I hope captures the sense of it. So in that same context, targeting the nucleus of the myeloma, as opposed to trying to keep the good guys in and cool the nucleus off, as it were, for want of a better term. The targeted cytotoxic space is very interesting right now because we have this agent melflufen, which is an aminopeptidase activated cytotoxic, and that delivers its payload right into the myeloma because myeloma is enriched for aminopeptidases, so basically cleaves the parent molecule, which in itself is actually quite lipophilic, which means it's delivered to the tumor cells that much better than other cytotoxics in the same setting, because those are typically actually lipophobic. Why is lipophilia important? Because in the bone marrow there's tons of fat, so you need to get your cytotoxics into where it actually is supposed to do its work. And the really neat thing about melflufen is it's designed to be more targeted in its effect, so it attacks the myeloma more and spares actually normal tissues to a greater degree. So that's a very exciting new area, and we've seen responses from that particular drug in a very important area of unmet medical need, which is extramedullary disease. This is disease that comes back typically after immune therapies have failed, and in that context it may be very difficult to treat. So I think for a variety of reasons, all of these drugs will have different roles, because we're recognizing that this isn't just a zero-sum game, that we need all of these approaches to confer benefit.